Welcome to our second Minimal Cell Workshop. I'm John, your host for today, um, tumor-free and ready to get going. So this is our second such event. I'm just curious. Um, I'm going to try to get where I can see on a different monitor how many of you there might actually be, but maybe that's not going to happen. I won't worry about it. So we were here about the same time last year. Last year we had 35 talks over 2 days. But the way I began last year is that before the before the JCVI minimal cell was available to the world, there had been almost 10,000 bacterial cell minimal cell publications. And based on work done at the JCVI over more than a decade, almost 20 years, we produced this in 2016. Clyde Hutchison was the lead author on a paper where where the Mycoplasma mycoides the Mycoplasma mycoides genome was reduced by a factor of 2 to yield JCVI SYN 3.0. And since then, we've now had over 32 publications using the JCVI minimal cell and 15 since last year's workshop. And I know there are a number of others in the works. So the great thing about when we produced this cell in 2016 is that once we had this cell available and the publication was out, folks from all over the world started contacting us, wanting the cell or wanting to work with us. Some people are working directly with the JCVI. Are other folks are just using it for their own purposes. Some people computationally, some people by wet science. But right now, there are more than 60 academic and industrial groups that I know of that are using uh, the JCVIV, which is, of course, the J. Craig Institute mentor, which is foolish. But there are 60 groups that have asked for the cell for us, or that I know that are working with us, and then I see from publications that there are other groups who are just using the cell based on digital information. Um, this is where we were in last year's slide for publications related to the JCVI minimal cell, but we've gone a lot farther than that. I look at it that there are a lot of reasons to use our cell, basically for investigating the first principles of cellular life. Our vision is that we would have a cell where every gene, the function of every gene is needed, and we know exactly what every gene and protein is doing. And we and others are developing algorithms for genome scale redesign so that we can build organisms to devote maximal energy to to specific purposes, and design microbes to solve human problems. Because it's incapable, because one of the cool things is because the minimal cell is incapable of infecting mammalian cells, we now envision using the JCVI minimal cell to investigate the pathogenic potential of proteins expressed by genetically intractable pathogens. And so we're going to have at least one talk that mentions this. An important thing, while our cell is not a primordial cell, we may have inadvertently recreated some of the features of primordial cells. So, for instance, the minimal cell 3.0 doesn't divide like a modern cell. It doesn't seem to form a protein lipid septum between two daughter cells. Rather, it looks like it may divide in the sense that a membrane vesicle containing what's inside a cell is now dividing based on membrane curvature. And so we identified seven genes that if you add back to the 3.0 minimal cell, it will divide like a modern cell. We also found inadvertently that we deleted the gene immediately upstream from bacterial histone-like protein HU. Now, normally cells would have about 10,000 copies, even mycoplasmas would have about 10,000 copies of this protein per cell. But by removal of the transcriptional control elements immediately upstream of HU, we went from 10,000 copies to only 28 copies per cell. And this may have accidentally recreated a cell that mimics what cells look like before the evolution of chromatin. We had a paper in Frontiers last year. I think Ben Gilbert was the lead author that in working with folks at Leiden University, Remus Dames group, in doing 3C, studies showed that the mycoplasma, the minimal cell, really did look different in this system than what you see with other bacteria. So I think that this is a remarkable feature of the minimal cell and deserves some more research. On day 2 of the meeting, I hope that we can have a discussion section where we deal with some of these issues. And this can happen again on Monday. Housekeeping, if you have issues, let's get in touch with Tim Stayes, and I will be trying to monitor how long people have talked. So hopefully we can go for 10 minutes.